Thank you. And thank you for giving me the opportunity to speak today. So severe IVH is the most common brain lesion in preterm infants and is associated with an increased mortality and a major neurological impairment. Over 50% of the infants develop a post-hemorrhagic ventricular dilation, PHVD, and requires the ventricular perinatal shunt. Despite a lot of research the last years, the complex cellular molecular mechanisms that causes the brain damage and the subsequent PHVD is still uncompletely understood. However, certain data have shown that there is a correlation between the blood volume and the damage. The development of the brain damage following IVH is associated with an increased inflammatory and oxidative response and activation of the periventricular brain tissue with a microglial and astroglial activation. It is well established that the pre-oligodendrocytes are extremely vulnerable to insults from oxidative attack. And this oxidative attack causes a maturational delay or a cell death to the pre-oligodendrocytes. In fact, research have shown that in animal models, by reducing the inflammation and oxidative insult, it has certain beneficial effects. Our research has mainly focused on the hemorrhage and more specifically on the hemolysis of the red cells and the subsequent release of the cell-free hemoglobin. Recently we showed in an animal preterm rabbit pup model of IVH that subsequent to the IVH there is an accumulation of methemoglobin, the oxidized form of hemoglobin, in the interventricular CSF at 72 hours of age. Furthermore, we show that there is a strong correlation between the level of methemoglobin and the inflammatory marker TNF-alpha. We were also able to show that in primary astrocyte cultures, we could cause an increased TNF-alpha mRNA and protein expression following exposure to methemoglobin. And in cerebrospinal fluid collected from preterm infants following IVH, we were also able to see a positive correlation between the TNF-alpha and methemoglobin. Thus, in the present study, we hypothesized that following IVH, the choroid plexus epithelial and the ventricular lining will be the first line of tissue structure that will be exposed to the hemorrhage and therefore will probably be very strongly affected by the hemorrhage. We further hypothesized that as there is an increased interventricular hemoglobin concentration, this will probably cause structural and functional damage to the choroid plexus and initiate an inflammatory response. More specifically, we aimed to try and characterize the causal role of cell-free hemoglobin, and we used again the rabbit pup model, which I will soon describe, but also primary human choroid plexus epithelial cells. Furthermore, we aimed to evaluate a positive protective effect of a hemoglobin scavenger, haptoglobin, in the animal model and also in the primary cells. So the study design was accordingly. We used the rabbit pup model of preterm IVH, which is well described by us and a group in the US. Using New Zealand white and loop rabbits and delivering the pups with cesarean section on day 29 with a full gestational day 32, we could induce the hemorrhage with a glycerol injection intraperitoneal at 2 hours of age. Using a high-frequency ultrasound, as you can see up here in this picture, we were able to verify hemorrhage in the pups. Using this high-frequency ultrasound, we were also able to, after randomizing the animals, inject intraventricular either a hemoglobin scavenger, haptoglobin again, or artificial CSF. at 6 hours of age. We then sampled choroid plexus epithelial tissue at 24 and 72 hours. Furthermore, we also used human primary choroid plexus cells and investigated the causal effects of hemoglobin exposure and protective effects of haptoglobin treatment. So, moving over to results. As can be seen here, we first investigated structural integrity of the choroid plexus using electron microscopy. And following IVH, a strong structural disintegration was seen here with disintegrated microvilli, apoptotic bodies, and swollen mitochondria as compared to control animals. Furthermore, we could show that there was increased inflammatory response as TNF-alpha, which is black dots here, as compared to control. And interestingly, treatment with haptoglobin resulted in a almost complete protection of the structural integrity and reduced inflammation. We then set on to further characterize the inflammatory response using mRNA analysis of the choroid plexus. And we were able to show that with potent inflammatory markers such as TNF-alpha, IL-8, MCP1, and IL-1-beta were all strongly upregulated following hemorrhage. So baseline in all these graphs represents control animals, and as you can see in the dark gray bars all over, there was a strong increase in the mRNA expression that shows fold change. Interestingly, treatment with haptoglobin resulted in a significant reduction in TNF-alpha, in IL-8, MCP1, and IL-1-beta, thus showing that scavenging of hemoglobin results in a reduced inflammatory response. In order to further see whether the cellular activation and intracellular signaling pathways that are responsible for this increase, we looked at again the mRNA expression and different pathways, and we were able to see that receptor pathways located to IL-1 receptor and the TLR4 system were strongly affected by hemorrhage. Again, the baseline represents a control animal, and we can see the IL-1 receptor increase, CD14, FAST, which is intracellular, and MyD88 is also intracellular signaling related to IL-1 receptor pathway and TLR4. And again, treatment with haptoglobin light bars could result in a strong reduction of the upregulated expression. Again, showing that scavenging hemoglobin affects this hemoglobin. We further wanted to verify that the increased mRNA expression seen could also be verified at protein level, and indeed we could see that increased protein expression of TNF-alpha following IVH, and treatment with haptoglobin resulted in almost complete reduction of the signaling. As I said, we further wanted to investigate the causal effect of hemoglobin in a primary human epithelial cell. So we used choroid plexus epithelial cells and exposed it to hemoglobin metabolites. And here we see endpoints of cell death, which is LDH release, and apoptosis, which is measured with space activation. And you can see that there is a strong effect on cell death and increased necrosis, but especially there is an increased apoptosis during the early hours. And very interestingly, methanoglobin effect was almost completely abolished with haptoglobin. So to summarize, we have been able to see that IVH is followed by release of cell-free hemoglobin and especially accumulation of methanoglobin in the interstitial space. IVH causes a structural and functional damage to the choroid plexus, and following IVH there is a strong increased inflammatory response in the choroid plexus. Interestingly, scavenging hemoglobin using haptoglobin seems to have very strong protective effects by reducing structural and functional damage, inflammatory response, and cellular activation. And in a primary human cell culture, we were also able to abolish these effects. So to conclude, hemoglobin may be an important causal contributor to the structural and functional damage which is seen following IVH. And removal or scavenging of hemoglobin using, for instance, haptoglobin or other molecules represent a therapeutic opportunity and efficient means to reduce the damage to the initial brain following IVH. I would just like to acknowledge some of the people that made this work possible, and especially David May and Sylvain Snipes, who were my collaborators in it.